Welcome to the first podcast on the urogenital system. And in part one, we're specifically going to be looking at the development of the urinary tract and urinary organs and glands that are involved in the production of our liquid waste. So as we begin, we go further back, again, back to our embryonic stage, and we are going to look at this tripartite series of kidney formation. We're going to start with a very early kidney in the cervical region. We're going to develop a middle, somewhat active, functional kidney. It's not going to persist. Neither of these are going to persist. And then the third kidney that we're going to form in our pelvic region, this is going to be our final adult kidney. So we're going to go through this progression. But first, we have to look very early in our embryo and look at this precursor tissue that is going to form from our intermediate mesoderm. So in the beginning of our urogenital system, as we mentioned, we are going to form the bulk of our urinary system and our genital tract and glands from our intermediate mesoderm. Remember, we have our paraxial mesoderm, our intermediate mesoderm, and our lateral mesoderm. Now, in this cross section, we can see that as we fold laterally and we create our gut, in this case we've got the midgut, along this abdominal dorsal surface, we're going to have this elevation of tissue. And if you're in the abdominal cavity and you see this elevation, you're going to recognize this as a urogenital ridge. So mesoderm has been proliferating quite a bit faster than the surrounding tissue. And we're going to have these paired structures on either side. Now, these are not just bumps, but these are long rows that run the length of this dorsal abdominal wall. And within this mesodermal tissue, we're going to create the nephrogenic cord, which is going to be the tissue that is all connected along this ribbon. On the inside, all of our nephrons, our tubules that are going to provide for the filtration and the production of urine. And on the external surface, more medial, we're going to create a secondary ridge of tissue out here that's going to give rise to our gonads. We can't see that quite as yet. So here in our video, we're starting again very early in embryonic development. We're at day 30, so we're still in week 3, essentially. But I'm wanting to remind you again about the 3 sources of mesoderm, the intermediate mesoderm, that being the source that's going to give rise to our urogenital ridge. Here's our lateral folding, and this is going to give us that cross-sectional view to allow us to see that abdominal cavity. Now, as you see the 2 white dots, there they are, the 2 white spots, that's going to be our intermediate mesoderm. We're going to see that's going to begin to protrude downward and create that urogenital ridge. The white could actually, you can almost think of that as being our nephrogenic cord, and the whole ridge, including the mesoderm on the outside, that being our urogenital ridge. Here's a nice way that you can look at how this ridge extends along the dorsal length on both sides of the embryo. The urogenital system can really be broken down into the urinary tract, which is the function of excretion, and the genital tract, which is for reproductive purposes, but we're also going to include the suprarenal glands simply because of their proximity and location to the developing kidneys. So we're going to start with urinary system. And with urinary system, we're going to see it develops first. And the urinary system is going to consist of these adult structures, our kidneys, which are going to function to produce the urine, the ureters, the transport conduit that removes the urine from the kidneys to the bladder is for temporary storage, and then at last for the expression of the urine through the urethra to the outside of the body. So these are the structures we're going to be developing in our embryo. Now, as we mentioned at the outset, our kidneys are going to go through a tripartite series of developmental structures. We're going to form first the pronephric kidney, or you can refer to it as the pronephros. And in this case, it is a rudimentary kidney. It is temporary, and it's often similar to the types that are found in primitive fish. It's not a very good kidney, in fact. Our second kidney, hence the name meso for middle, the mesonephros, this is going to be fairly well developed. It's temporary in humans, but in amphibians it's going to persist quite a bit. You've seen this mesonephric type kidney if you've had comparative anatomy and looked at the mud puppy. Lastly, the metanephros, that's going to be our permanent adult organ. And so they're going to form in sequence. There's going to be some overlap, but you'll see the pronephros first, the mesonephros, and then lastly will form the metanephric kidney. So in this illustration, we can see in a sagittal view and more of a coronal view what this is going to look like. Here in the cervical region, we have the pronephros, and it's somewhat dashed. And you can see a few little circles on the inside because, again, this is very short-lived structure. Then we have the mesonephric kidney, which you see here in purple. And then this is the very beginning of the formation of our metanephric kidney. And again, a cross-section, you can see this diverticulum that's going to be forming here. That is going to form our metanephros. There's our mesonephros. And then our pronephros is degenerating in this particular illustration. So the pronephric kidney, this is the first set to form. Again, early 4th week, not a surprise that we see this. It's going to consist of a few cells and a few tubules. Now, when we talk about tubules, think of these as very primitive rudimentary nephrons. We're not actually giving them that name yet. So we just call them urinary tubules. And again, these are in the cervical region. But the most important thing, in the cervical region, you start to form a duct. And this duct is going to connect these tubules to the cloaca. And so since you're servicing the pronephros, this duct is referred to as the pronephric kidney. The pronephric duct. Now, this pronephric duct, the part associated with the cervical pronephric kidney, that's going to persist. But the remainder of the duct is not going to degenerate. It is going to remain, and it is going to be used by the next 2 stages of kidney development. It's going to be used both by the mesonephros and the metanephros. So the pronephric duct is going to persist by and large. Only a small part's going to degenerate. And so we can almost think of the pronephric kidney as being a space holder or maybe an inductive center to help form the proper location of this all-important duct that's going to be recycled through the remainder of our urinary tract. Now, the second kidney to form, the mesonephros, this is late 4th week, and it's often referred to as the interim kidney. Now, in this case, it is a functional kidney because it does have glomeruli and it does have these mesonephric tubules. Again, think of the mesonephric tubules as primordial or very primitive rudimentary nephrons. They will have Bowman's capsules around the glomeruli. They will have convoluted tubules, but they're not going to be exactly the same as the nephrons we see in the metanephric kidney. So, for example, here in these cross-sectional views, here we can see our urogenital ridge. We've got our nephrogenic cord working here. Since this is a transverse section, we're cutting our mesonephric duct. Remember, this was the former pronephric duct. We just simply rename it. And we have this mesodermal tissue. At first, we call it a mesonephric vesicle. It's going to fuse with the mesonephric duct. Then you can see we also have blood vessels growing away from the dorsal aorta, forming a glomerulus. And this mesoderm is also going to become associated with the glomerulus, and we have this mesonephric tubule. Again, here we can envision Bowman's capsule. Here we can see a convoluted tubular portion tying back to the mesonephric duct. You can almost think of that like a collecting duct. So in many ways, this does look like a nephron, but for our mesonephros, we're only referring to this as a mesonephric tubule. Now, since this is interim, and we're going to replace it with a metanephric kidney, what we are going to have in many people, and certainly in males, we're going to recycle some of this tissue. So the mesonephric tubules that we have in the mesonephric kidney, these are going to persist and be recycled and form the efferent ductules of the testes. And when we get to that, we'll talk about that a little bit more. But the efferent ductules are the structures that are going to drain the spermatozoa from the testes proper and empty into the epididymis. It's our first extratesticular duct. The epididymis then is going to contact with the vas deferens that then ends up with the urethra. So we are going to recycle these. And what we're going to see is that the mesonephric ducts may also be utilized for various other adult derivatives in males, and we'll talk about that when we get to our genital tract. So now let's look at a very short video clip that's going to show you and introduce you to the concept of the changing from the pronephric kidney into the mesonephric kidney. Now, the thing that I didn't particularly care for in the video is the pronephric and mesonephric duct. It appeared in the video that it was being slowly drawn in as you're developing the mesonephros. The duct is already there, so that's the only artistic license that I have a problem with in that video. Otherwise, that's a decent little video. We've got the pronephros that's degenerating. The mesonephros is working. Well, by the late 5th week, we begin to form our permanent set of kidneys, the metanephroi, which is plural. Metanephros is singular. Metanephric kidney or kidneys is another way that you can also say that. But we are going to be functional about 4 weeks later. So 9-10 weeks, we're going to be producing urine. And this is going to occur throughout fetal life. And so your amniotic fluid serves a lot of roles, and not the least of which it is your sewer while you are in the womb. Now, the kidneys are going to develop from 2 different types of mesodermal tissue. Let me just make that case up front. These are both mesoderm. And so early in one of our images, we saw what was labeled as the uteric bud, the beginnings of our metanephric kidney. Another name for uteric bud is the metanephric diverticulum. And remember, a diverticulum is a blind-ended pocket. This is going to form much of the passageway of urine from collecting tubules all the way down to the bladder. That portion of the kidney, the urethra, that's going to come from the metanephric diverticulum. The metanephric mass of intermediate mesoderm that's going to be induced to form by the uteric bud. This is going to create the nephrons and all of our cortical labyrinth that we have inside the kidney. However, both of these are mesodermal in origin. So here we can see low down on our mesonephric duct, we're going to have the branching, and this is going to be the uteric bud, the metanephric diverticulum. And we can see how this extends out. It's going to induce the mesoderm surrounding it, this intermediate mesoderm, and that's going to become the remainder of the kidney. So this is going to extend out. And as it grows larger and larger, this diverticulum is going to transform into a stalk, and we call this the off of the metanephric duct. And this stalk then, the diverticulum, as I mentioned, it's going to give rise to the ureter, the renal pelvis, the major and minor calyces, and collecting tubules. However, this uteric bud, as I've mentioned, this is going to be inductive in nature and cause the surrounding mesoderm to form the cortical labyrinths and all the connective tissue of the kidney. So as we extend further, we can have the branching that we see here of the uteric bud. The first generations are going to be the major calyces, as we extend further out, and we'll see this in a video that's coming up later, the next generations of branches, these are the minor calyces, and then we're going to have these branching collecting tubules that extend up into the cortex of this mesoderm that is forming here. You can see the little florets that are beginning to form, these clusters, these mesenchymal clusters. These are going to be mesodermal vesicles, and it's going to be from these that we begin to form. Much like we saw with the formation of our tubules, these are going to form our nephrons and the cortical labyrinths. So as we get to these straight collecting tubules, portions of these tubules are going to branch, and you can see the branching nature here. And at the tips of these branched portions of our collecting tubules, this is where we're going to induce that mesoderm to begin to condense, and it's going to form a vesicle. This is a metanephric vesicle. And the metanephric vesicle is going to continue to grow and expand and lengthen, and this is going to become a metanephric tubule. Now, these tubules would be somewhat synonymous with our pronephric tubules, our mesonephric tubules. And the thing that I want you to notice is, much like the youngest part of a tree that is growing is going to be the top part. In this case, the youngest of our nephrons are going to be those nephrons that are closest to the outer capsule. The oldest are going to be those deeper, and these are going to be juxtamedullary. They're at that boundary between the cortex and the deeper medulla of the kidney. Because you can see as the straight collecting tubule continues to grow and branches again, the tips of these branches are going to induce another set of our metanephric vesicles, and that's going to continue for a number of generations. So the straight collecting tubules branched, the branches induced the mesoderm to form a metanephric tubule. And now as these grow and extend, we're going to see that they come into proximity with blood vessels, and so they are going to be invaginated with a capillary bundle that's going to be the glomerulus. And so this is going to create our renal corpuscle. Remember, the renal corpuscle is Bowman's capsule and its associated glomerulus. And now we can begin to recognize the different components of our cortical labyrinth: proximal convoluted tubule, loop of Henle, distal convoluted tubule, and finally collecting duct. So that's going to comprise our nephron, the renal corpuscle, the different convoluted portions of the tubule, and our loop of Henle. Also recall from phys and from histology, a nephron plus its collecting tubule is referred to as the functional unit of the kidney, that being a uriniferous tubule. In this final illustration, once again, bring the point home that the oldest, most developed of our nephrons are going to be the ones that are deepest. They're going to be the ones at the juxtamedullary boundary. And the youngest, least developed, are going to be the ones closest to the capsule of the kidney. Now, here's a video showing the development of the metanephric kidney. The metanephric kidney, as we recall, forms in the pelvic region. And really, when we think of the kidneys that we have today, it's more of a lumbar at best region. We're not very low in the pelvis. In the case of the embryo, we're down in the very caudal end of the embryo. And so the kidneys are going to relatively move. And so what really is going to happen is the embryo is going to grow away from the kidneys. And there, if you could anchor the kidneys, you could see the floor of the pelvis begin to drop. And in doing so, you have this relative ascent of the kidneys upward in the abdomen. Now, as you do that, the kidneys are going to have a rotation, and they're going to have a series of arteries. that are going to be supplied to the kidneys to help connect the kidneys back to the dorsal aorta. As new ones are formed, old ones are going to degenerate. In this video, the relocation of the kidney, this is explaining what we're talking about. We can see the metanephric kidney low in the pelvis. Now, as you see the embryo growing, here's that apparent movement. The kidneys are going to rise upward, and they're going to rotate 90 degrees medially because they were facing, the hilum was facing toward us. There you can see the series of arteries that grow in and the arteries that degenerate as the kidneys ascend up to the point of the suprarenal gland. To supplement the video, here we can see you almost think of this as a stop motion animation showing the kidneys in the lower pelvic region, and then as they rise, they maintain their connection back to the common iliac artery. Continue to go higher, we degenerate that first artery for a second that is grown in, and then continued ascension, we're pulling along our ureters with us, they're extending in length, and we lose the second set, and there we have our third set. That's why it's not uncommon to see a kidney with more than one renal artery. Now, if you do have more than one, unfortunately, those are not accepted for organ transplantation, but it's not an uncommon thing to see. It's simply because one of the earlier embryonic vessels simply didn't degenerate as it should. Now we move our attention to the urinary bladder. Now, the urinary bladder is going to form from a structure that's called the urogenital sinus, and we'll see a picture of that in just a moment. But the urogenital sinus is going to consist of three portions. The cranialmost portion, this is going to be continuous with the allantois, so we're here in the foregut region. This vesicular portion is going to become the bladder. The middle portion of our urogenital sinus is called the pelvic portion. It's going to become the urethra in females, and specifically the prostatic urethra in males. And then lastly, this most caudal portion of the urogenital sinus, is the part that's closest to the cloaca, is considered the phallic portion, and it's going to grow toward and become a part of the genital tubercle, which is going to be the beginnings of the formation of our external genitalia. Here's our illustration. Here we can see the allantois. It's part of our connecting stalk. This is going to be the vesicular portion that gives rise to the bladder. Here we see the phallic portion and the pelvic portion in the middle. You can see that even more easily illustrated here. Now, here is our hindgut, and our hindgut is being separated from this portion that's consistent with the allantois by this column of tissue. This is called the urorectal septum. This tissue is going to slice down, and it is going to separate our hindgut into these two separate areas. This is going to become our rectum. It's going to fuse and form the anus for our solid waste removal, and this is going to be associated with our urogenital tract. And we're going to completely separate those as the urorectal septum continues to move toward the cloacal membrane. Now, back to our discussion about all the derivative tissue. The lining of the bladder, since we are talking about allantois hindgut, this is endodermal in nature. The connective tissue, the smooth muscle, remember that's going to be splanchnic mesoderm, not unlike what we've seen in other organs, certainly in respiratory tract and in urinary. Now, the allantois, portions of the allantois, the most distant portions, these are going to degenerate, and all that's going to remain is a fibrous cord that's called the urachus, and this is going to extend from the tip of the bladder toward the umbilicus, or your navel, and that's what's going to remain. Now, this urachus in the adults, it's going to persist, and it's referred to as the median umbilical ligament. So when you get to gross anatomy, the median umbilical ligament is formerly the urachus and formerly the allantois during embryonic development. Now, we mentioned how some of the mesonephric portions of that interim kidney are actually recycled and reused. Well, in this case, what we're looking at is a split open portion of a bladder, and this is from a male. We can see the prostate gland here at the bottom. And so this is going to be our prostatic urethra. This is a slice through the penis, actually. So here's our prostatic urethra, and here our urethra is entering into the bladder itself. Now, where we have the orifices of the ureters on both sides, and then the opening of the urethra, that is what's referred to as the trigone of the bladder. Now, this trigone of the bladder is created because of the connection of the mesonephric ducts, which are going to be our ureters, and the urethra. But when we incorporate that tissue, that is going to help create this structure that is known as the trigone. Some of that tissue is going to be recycled and utilized in the formation of this portion of the bladder. We're going to see a similar thing happening. Embryonic structures are going to be incorporated into the developing heart as well. So this is one of these regions where we do that. We're also going to see that in males, the caudal portions of the mesonephric ducts, these are going to become the ejaculatory ducts. And we'll see when we get to our genital tracts, we'll see exactly where these are located. But again, we're continuing to recycle parts of these mesonephric ducts. Remember, our mesonephric tubules we already recycled into our efferent ductules of our testes. In females, this caudal portion of the mesonephric duct is going to degenerate. Now, that is a common theme that we're going to see. The mesonephric duct will persist in males. It will degenerate in females. We have a second duct that we're going to talk about when we get to genital tract. That's just the opposite. It will degenerate in males and persist in females to provide them for their genital tracts. Now we have a short video of the bladder. And now to complete our discussion, we are going to talk about the suprarenal or the adrenal glands. We can see that the adrenal gland is comprised of the cortex and the medulla. The cortex being mesodermal and the medulla being derived mostly from neural crest cells. Now this is important because these neural crest cells in the medulla, they are in fact going to give rise to some of our sympathetic ganglia, specifically our chromaffin cells, that are producing a lot of epinephrine and some norepinephrine. So they actually function as postganglionic neurons. So we're sort of giving them this honorary distinction as sympathetic ganglia. So neural crest cells that give rise to a lot of nervous system tissue are in fact the precursor tissue to give rise to these chromaffin cells that are present in the medulla that produce the epinephrine. Now, the mesodermal cells for the cortex, we're going to see a lot of those coming from the mesothelial or mesodermal lining of the body wall that is simply incorporated along with these medullary cells. Now, we have a single medulla filled with chromaffin cells, but the cortex is going to break down into three distinct layers. The outer layer is the zona glomerulosa, the middle layer is the zona fasciculata, and the first layer is the innermost layer is the zona reticularis. Now these have histological distinctions. They're also going to have functional distinctions in the sense of the hormones that they produce. Some are going to be mineralocorticoids, some are going to be glucocorticoids, and some are going to be androgens. So these hormones have a great impact on metabolism, on the retention of water, and certainly on the development of masculine and feminine secondary characteristics.